Okay, so first, when we talk about antibody, they are globular glycoproteins. Globular glycoproteins. So they are protein, basically. When we talk about glycoprotein, they are protein. Okay, and in fact, they are quaternary proteins. Okay, they are quaternary protein because they contain consist two identical short polypeptide known as the light chain and two identical long polypeptide known as heavy chain. Are you clear? Okay, so we have the antibody now consists of four total. You can see that 4 polypeptide chains. Okay, understand? So it means that it's a quaternary protein. Okay, when we talk about protein, so level 1 or we call it primary, secondary, tertiary, only 1 polypeptide. So when we talk about quaternary protein, means that they have more than 1. So in antibody, they have 4. Okay, and then if you look at this, they have what we call the light chain because they're shorter, and also heavy chains, which is longer, and they're identical. Okay, so when you talk about globular basically means that you have to understand they are water soluble. Okay, with the hydrophilic R group pointing to the surrounding. Okay, so this antibody, also known as the immunoglobulin Ig. Okay, immunoglobulins, and they are divided into 5 subclasses. Okay, total we have 5 subclasses. Okay, so the 4 chains, okay, in the antibody molecule, they are held together by 4 disulfide bonds. 4 disulfide bond. Okay, so again, these disulfide bond actually hold these 4 polypeptide chains together, forming a Y-shaped quaternary protein. As I say that they're quaternary protein, and each chain will have the constant regions and variable regions. Okay, so now I won't go into the detail about constant change and variable change. Let us look at the diagram here. In activity 12.6, you can take a photo or you can actually draw it out. So when you draw it out, you can see that each antibody molecule, okay, they will contain a long chain, which is we call it a heavy chain. Okay, so I label here. So this is a heavy chain. And also you can see that 2 light chain or short chain. Okay, so this one we call as the light chain for shorter. So each change, you can see that the lighter regions here, the lighter color actually indicates that they are the variable regions. Are you clear? Variable regions. Okay, and then the darker regions here is known as the constant region. Are you clear? So basically, this part is called variable regions. The remaining part actually is known as the constant region. So means that as long as they are from the same subclass. Now, antibody molecules, we can divide them into 5 subclasses. So these 5 subclasses is named as IgG, IgM, IgA, IgD, and IgE. So there are 5 subclasses in total. Are you clear? So if this antibody is classified as subclass IgG subclass, if IgG, so means that all these antibody, their constant regions will be the same. Means that amino acid sequence will be the same. What make each antibody different is only the variable regions. Are you clear? Variable regions. So each, guys, can I see that? Huh? Now, each antibody molecule will have 2 antigens binding site. Are you clear? We have 2 in total. So another one's here. Okay, antigen binding site. Are you clear? So we have 1 antigen binding site, 2nd antigen binding site. But what is FAB? fragment. FAB actually stands for the antigen binding fragment. So where is antigen binding fragment? So this is antigen binding fragment. Are you clear? So total we have 4 antigen binding fragment. Because we have 4, can you see that? Antigen FAB, antigen binding fragment. We have 4. Are you clear? Okay, then these antigen binding fragments, they are different for each antibody. Are you clear? Each antibody. So later I will show you a few examples, then you'll be able to understand. Okay, more. Now, each antibody also have what we call the FC regions. Okay, so this is the FC regions. C stands for constant. It's a constant fragment. Are you clear? It stands for constant fragments. So total we have 2 FC. Are you clear? 2 FC fragments. So these FC fragments will allow us to determine which subclass of the antibody. Are you clear? It belongs to which? Okay, so we did because all IgG will have the same FC region, all will have the same, IgM will have the same. So therefore, this can be used for us to detect, oh, this IgG, this IgM. So we call FC. Okay. So these 4, can you see that these 4 change 1, 2, 3, 4? They are linked by the disulfide bonds. Okay. So you can see a total we have 4 disulfide bonds. And another last part, very important here, we call it a hinge region. Hinge region is something like a spring. Can you see like a spring there? So give us the flexibility. So what's mean by give us the flexibility? So if you try to imagine, if let's say I have the antigens. So these 2 antigens, they are far away like this. So means that the same antibody actually can open up large enough, long enough, distant enough to bind. Are you clear? So 2 antigen binding site. So if these 2 antigens, they are very, very near, let's say, doesn't matter, your antibody or our antibody still will be able to bind because these hinge regions give the flexibility. Okay, clear? So you can take a photo if you need, or you can actually draw it out because in the exam, say you might be required to draw. Okay. Yeah. Clear so far? Okay. Now, if no questions, then we continue with this. Okay. So constant region. What's a constant region? So in the constant region of the heavy chains, the amino acid sequence is constant within a particular Ig class or subclass. So the C regions of the heavy chains is the tail, okay, of the Y parts of the antibody. So this is the constant regions. Can you see that? The tail here, the constant region, the tail, okay, generally. Okay. Variable region. Variable region has a unique amino acid sequence because this sequence will allow the antibody to fall into precise 3-dimensional shape, forming antigen binding sites, allow it to bind with a specific antigen. So this F, okay, here we have a specific sequence. Okay, it can be amino acid 1, 2, 3, 4, 5, and then another one is 5, 2, 3, 2, 1. Okay, so therefore it will give us this shape, but they're identical. Are you clear? They are identical. Yeah, identical means what? This part and this part identical, this one and this one identical. Okay. Now, in the Y shape of antibody, the tail of Y is known as FC fragments. Can you see that? The tail here is called FC fragments. So the FC fragments bind to the receptor on the phagocyte and other cells of the immune system. This one I will show you guys later when we talk about the role of the antibody in opsonizations. Okay, so this region of antibody molecule also bind with the molecule of the complement system Now, what is my complement system? Complement system actually can help to kill the pathogen. Okay, but the question here is complement system will attach to the FC regions or FC fragments of the antibody. Against these two, at this moment, you may not be able to see clearly, but it doesn't matter. I will tell you guys more when we come to the this part. Okay, so the two arm of Y are referred as FAB fragments. These two arm FAB fragments. Okay, so the tips of the FAB fragments bind to the antigen. So at the tips here, you can see that it's not entire thing. It may not be entire thing. It's just these tips. Right? At the tips here, bind to the antigen. So the Y shape permits the antibody molecule to combine with two antigens molecule, okay? Two non-self antigen, which allow forming this antigen antibody complex. It's like enzyme substrate complex. Enzyme, substrate bind to enzyme. So we call it enzyme substrate complex. In this case, antigen antibody complex. Okay. So the hinge regions, as I say, that give the antibody molecule flexibility. Okay. So what are the classes of antibody? What are the classes of antibody? So in mammal, okay, it depends. In mammal or in other, different type animal, they may have different classes of antibody. But for us, so the different classes of heavy chain that form the 5 classes of immunoglobulins, they are IgM, IgG, IgA, IgD, and IgE. Okay, oh, sorry, I shouldn't highlight all. So now, normally it means our focus would be on IgG. Okay, why we focus on IgG? Because IgG involved in the passive immunity, which we are going to talk about this in the next subtopic. IgM is the one that involved in the agglutination, clumping, okay, in general. Okay, so the rest are not really, actually important, okay, but at least you know about IgA, IgD, and IgE. So before we go into each subclass and attempt the activities here, let us look at this in a little bit detail. But again, as I say that this is not in our syllabus. Okay, it's not in our syllabus. So therefore, I will go quickly. I'll tell you guys about IgG. So IgG, as I said that just now, I highlight IgG. So why I highlight IgG? Because the next subtopic we're going to talk about passive immunity or natural passive immunity. What's so special about IgG? IgG, if you compare the molecular weight, you will realize that, can you see that? You'll realize that IgG actually is the smallest one, and most importantly, they actually can cross the placenta. They are so small, they will be able to cross placenta. So if they cross the placenta, means that it can be received by the fetus, and also, again, it can be received by the fetus. Therefore, it gives the fetus and newborn baby what we call the natural passive immunity. They are very, very small among, all of them. It's about 150,000 Daltons in this case. Okay. So don't worry about the complement fixation. Complement fixation. Okay, don't worry about this. IgM. As I asked you guys to highlight IgM. IgM, why we talk about it? Because in the functions later, in the functions of the antibody later, we are going to look at this, what we call the agglutination, okay, or agglutinations process, where you can see that they can bind with many antigens, hold the bacteria, hold the pathogen together so that they won't be able to spread to other place. Are you clear? Okay, so agglutination, clump. Okay, another word is called, layman term is clump all the pathogen together. So this is IgM. IgA, not so important, but we may want, on a later stage, on the next subtopic, we also will talk about it in terms of the natural. Okay, write down. Okay, here, natural passive immunity also. But not for fetus, because it cannot cross. You can see the molecular size is so big, they cannot cross the placenta. Definitely, they cannot cross the placenta for IgA. Okay, but they appear, this IgA appear in the milk. Okay, so this is very good for the newborn baby, the breastfeed. If the mom breastfeeding the kids or the baby, what we have here, the IgA in the milk, IgA in the milk will help to coat a layer to protect the mucosa surface. So means that they were located at the mucosa, means that the intestinal mucosa, okay, GIT mucosa surface. Mucosa surface means the surface of the mucus, okay, the surface of, the surface of the cell that can produce mucus. So they always coat it. So means that the pathogen won't be able to colonize the intestinal tract of the baby. Okay, IgD, so far we do not know what is the serum function. Means that generally they are the B cells receptor. We learn about B cell receptor. So B cell receptor actually comes from IgD. Are you clear? B cell receptor always come from IgD. So that's why they don't have the serum function. We don't secrete IgD out, because IgD actually involved as the B cell receptor, always anchor on the B cells. IgE slightly, you can see that it's slightly larger compared to the IgD. So they are mainly involved in the allergic reactions, okay, and also the parasitic worm infections. Okay, so for us, I say that IgM enough already for us to know. Okay, so you can actually take 1 minute or 2 minutes to continue with the activity. Did you know what 12.3? Okay, so the first type of antibody to be secreted during the primary response always start with generally IgM first. Why? Because they form this agglutination process. Okay, they form this what we call the agglutination. So when you see agglutination clump, this pathogen all together, because we want to reduce the mobility, we don't want them to move further away from the infected site. Are you clear? We try to catch as many as possible. We don't want them to go to other side, for example, enter into the blood and cause the sepsis. Sepsis basically means that the blood contaminated with the bacteria. Because if the blood is contaminated with bacteria, this bacteria can move to any organ. It will cause the organ failure. Okay, so therefore, generally in primary response, we always detect IgM first. Are you clear? IgM. And then when the secondary response, that is where IgG will be secreted. Okay, IgG will be formed. Are you clear? And this IgG, the good thing about IgG, they can cross the placenta, because right now they are the smallest immunoglobulins antibody. Okay, they are smallest. So therefore, they can cross the placenta and protect the fetus and later newborn baby. So this one we say that is a natural passive immunity. So next subtopic, we're going to talk about this natural versus artificial, passive versus active immunity. What are their differences? Okay. Now, so it's important immunoglobulins on the B cell surface. So it's critical role in maturation of B cells and help activate B cell following antigen binding. So basically, in this case, they act as what we call the B cells receptor. Still remember, during the maturation process in the bone marrow, the B cells actually develop B cells receptor. And I say that this B cell receptor actually, they are the antibody. So this is actually IgD. Are you clear? So in terms of the plasma, in the blood plasma, they have very, very low concentration. They don't have much role if you compare to others. Okay. So it is present in mucus, breast milk, highlight breast milk, and particularly prevents the viruses and bacterium from attaching to the epithelial surface or mucosa surface. So this one actually is give us what we call the natural passive immunity. Okay, it gives us a natural passive immunity. So IgA. Okay, so from the breast milk. Now, this form of antibody promote the release of the histamines involved in the allergic reactions, parasitic worm infections. So this one is IgE. Okay, IgE. So for us, the most important one, if I ask If you ask in the questions, generally these two are more important. And if let's say the third one, I will say this one, okay, because it involved in the next subtopics, okay, natural passive immunity, involved IgA. Okay, so with this, I have done for the subclasses. Let us continue to the next part, the mechanism of the antibody actions. Okay, how antibody actually involved in the immunity or immune response. So generally, you have to understand antibody, they do not actually kill pathogen. They don't actually kill pathogen, but they bind to the antigens only. So by binding to the antigen, they interfere with the pathogen's activities, or they mark the pathogen in various ways for the inactivations or destruction. So antibody, always remember, they don't kill. But one good thing about this, we want to interfere with the pathogen activity and also mark the pathogen. So what does this mean? Either toxin, pathogen, they always interact, okay, in our interact with the cell surface membrane, so that it will cause the reaction take place. If let's say, guys, this is our host cell. So host cells will have the cell's receptor, okay, cell surface receptor. Not for virus, not for the pathogen or bacteria. It may for signaling molecule. Let's say signaling molecule. So signaling molecule can bind to this binding site. But it so happened that the pathogen may have the same, geometrical, what we call the antigens, we have the geometrical shape, also can bind. Can I see that? So this is signaling molecule. This is the pathogen. Can you see it? So these pathogens also have the non-self antigen that can bind, and it will cause the for example, it may actually cause the cell to be infected. Are you clear? Cause the cell to be infected. So what we're going to do here is, before this pathogen infect the cells or enter, invade into the cells, we actually label them, bind them with the antibody, for example. Y-shaped antibody. Can you see that? So it means that now this antigen won't be able to interact with the receptor. So we interfere the pathogen activity. Are you clear? And at the same time, we also mark the pathogen so that we can go, we can inactivate it. Okay? For example, we can clump all of them together so that they cannot move away and go for destruction. So destructions here, okay, involve two systems, either by, right now, by complement system, okay, or by phagocyte. Okay, by phagocytes, right? The destructions. So let us look at one by one, the mechanism of antibody actions. Okay, so the first one, look at this. So bacteria, 1, 2, 3, we clump them all together. So this process is known as agglutination. Okay, so basically antibody with multiple antigen binding site, particularly IgM. Okay, IgM. Particularly, okay, can cause the agglutinations clumping together of the bacteria, reducing the chances of spread throughout the body. And also when you agglutinate them together, they are large, so they can be detected by the phagocyte. Are you clear? They are large, so it means that you clump them together. Okay, it's similar to when next time when you work, okay, let's say for example, you want to take off, okay, one person go out from the office, no one know, okay, but the whole gang go out from the office, people will know, right? Okay, you actually skip a work, right? So it's something like this. So our defense, our immune cells will be able to actually notice it, okay, because clump. Second, complements. So activations of the complements. So what's the activation of complement? Now, let's look at this. In terms of activation of complement, is like this. Our antibody, okay, carry a toxic substances or the protein, they will cause the lysis of the cells. So it carries this. So this is termed as complement. So complements actually have the ability to lyse the cells, okay, the pathogen. Are you clear? Okay, the complements, okay, this complement. So complement is just attached. It's not part of the antibody. Okay, they are not part of antibody. They are just attached. So when you learn antibody, so antibody won't kill, but its complement system is the one. So when this, okay, pathogen. So pathogen have the cell surface antigens, right? So when this activated, when this antibody bind to the antigens, what will happen here? So this complement system is activated. So complement actually we're going to lyse the pathogen. Are you clear? So once become activated. So can you see that? They bind to the antigen, therefore release, okay, the toxic substances, particularly the complex that can form the pore in the foreign cell's membrane, allowing water molecule and ions to rush in. So therefore the cell will swell and eventually will lyse the cell. So this is what I call activations of the complement. Against antibody just help to mark. They don't actually, they cause the killing process. The killing process is done by the complement system. Okay. Now third, we look at this called opsonization. So what I mean opsonization? Opsonization basically is the binding of antibody to the antigen on the surface of the bacteria, promote phagocytosis or enhance the phagocytosis by macrophages and neutrophil. So now what does this mean? Let us look at opsonization or enhanced phagocytosis. Label and enhanced phagocytosis. Now what actually happened here is I have the pathogen. So with the cell surface antigen. Okay, so I have the macrophage. Okay, so this is the pathogen. So macrophage, they are phagocyte. Are you clear? And they involve in non-specific. They involve in a non-specific, okay, defense mechanism. So means that if a non-specific means that they can't or they cannot recognize the antigen. Are you clear? So therefore, in order to increase, okay, in order to increase the phagocytosis, then we perform what we call the labeling by antibody. So how this thing can take place? So at the cell surface of the macrophage, how to do this? Okay, so first of all, look at this diagram. So I have pathogen. So this pathogen with the, what we call this cell surface antigen, non-self antigen. So in order to enhance the phagocytosis, doesn't mean that macrophage cannot carry out phagocytosis, but we want to enhance it, feel better, faster. Now you try to imagine, if I try to, you try to imagine, okay? So you are an army, for example, you have a gun and you want to shoot the enemy. But at this moment, you are not sure who is the enemy, who is not. Is it maybe there are civilians? This one is the soldier, means the enemy. These are civilians, okay? So you don't want to open fire and then you killed the wrong one, correct or not? So you take time to think, observe, think, long, take long period of time. But now, if I tell you, civilians, they wear blue color shirt, enemy wear red color shirts. So we can label them immediately. You do not need to think, red color, I shoot, I kill. Red color, I shoot, I kill. So how to help in this case? Antibody. So antibody, we're going to bind to this, even label, because we know that under normal conditions, okay, antibody only bind to the non-self antigen. Are you clear? Okay, so what actually happened now? If a label like this, okay, now phagocyte, for example, macrophage. Macrophage actually, they do have what we call the FC receptor. They do not need to recognize. Means in this case, guys, the army, you do not need to know, oh, this enemy from Germany, this is enemy from China, this is enemy from US. No need. Because as long as they wear red shirt, you know that they are enemy. Cannot. So as long as the pathogen, as long as the cell is labeled with the antibody, we know that they are pathogen. They have non-self antigen. So what happened here is macrophage now extend the cytoplasmic projection. Okay, they extend the cytoplasmic projections, for example. And start to engulf the pathogens. How they can do this? Because at the surface, okay, draw it better, sorry. At the surface of the macrophage, they do have a special receptor here known as FC receptor. Okay, label. This one is known as FC receptor. So FC receptor, we're going to recognize the FC regions of the antibody. Are you clear? So they bind the FC regions to take the cells in, okay, by phagocytosis. So enhance the phagocytosis. Are you clear? So phagocyte, no need to understand, oh, this is the pathogen 1. Oh, this is the pathogen 2. This is pathogen 3. Do I need? No need. As long as they are labeled with the antibody, we can kill them already. Are you clear? Can you see that? We can kill them already. So we do not need, again, to carry out phagocytosis, we do not need to know who are them. Are you clear? Okay, so this FC receptor, then phagocytosis can take place. Okay, so if you need, you can take a photo of this, what means the opsonizations, okay? Okay, the next one is a little bit complicated because it's called antibody-dependent cell-mediated cytotoxicity, or ADCC. Okay, now this one you no need to worry because this one is not in our syllabus, for information only. Okay, so what actually happened here is similar, right? But involve eosinophil. It may involve some natural killer cells, monocytes, macrophages. What they do actually, again, they do have a receptor known as FC receptor. Still remember? Okay, they do have this FC receptor. So this FC receptor can recognize the FC regions of the antibody. But this one generally involve larger target cell. Okay? For example, parasitic worm. Are you clear? Parasitic worm. So what happened here is this eosinophil contain the porphyrin. So remember porphyrin? Porphyrin, we're going to make pore and also the hydrolytic enzyme to kill the parasite. But the thing here, eosinophil have to know where are them. So it means that eosinophil will have this FC receptor bind to the antibody. Are you clear? The IgE, yes, still remember IgE just now? IgE. So that's why they know how to bind. When they bind, they will release the toxic substances and then kill the parasite. So this is called ADCC. Okay, antibody-dependent cell-mediated cytotoxicity. Can you see that? Antibody-dependent, we depend on antibody. Cell-mediated because we still use the cell to kill the parasite. Okay. The last one you need to know, highlight, is a neutralization. Neutralizations. So basically, just now I said that viruses, bacteria, toxin, they can interact with the host cell surface membrane and give us the trouble. Are you clear? Okay. So what we can do, we're going to neutralize them. Basically, how to neutralize them? We get the antibody to bind. So now can you see that initial shape, which is a round shape like this, but now the shape is like triangular. So they no longer can interact with the, okay, they are no longer can interact with our cell surface receptor. Therefore, it won't be able to invade into the host cells. So for example, antibody bound to the antigens on the surface of the virus, neutralized by blocking its ability to bind to the host cell's surface receptor. Antibody can combine with the toxin, neutralize them, and making them harmless. Okay, so this kind of antibody known as antitoxin. And antibody can attach to the flagella of the bacteria and also making them less active and easier for phagocytosis to engulf them. Okay, so with this, I have done for the antibody and the role of the antibody.